That's you. Окей. Значит, мы продолжаем наши публичные лекции. И вот это, значит, вот эта и следующая лекция будет прочитана нашими гостями, которые работают вместе с Аней Торгашевой в нашей лаборатории по ее гранту на приглашение ведущих ученых, на совместную работу с ведущими мировыми учеными. Так что я рад вам представить двух ведущих мировых ученых. Two leading scientists who kindly decided to leave the comfortable and nice countries and move to bloody Siberia to work with us. And so I'm happy to introduce Alex Su from University of Uppsala, and now he is moving to University of East Anglia, and Francisca Ruiz Ruano, he is from the same laboratory, and he came to us from Peru. Yeah. Yes. So today we have a lecture of Alexander and about this DNA elimination, and next week we shall have a lecture of Francisco about satellite DNA is a toolbox for cytogenomics. Okay, Alex, thank you very much that you came to us.
Добрый день, everybody. That's as far as my Russian goes, I'm sorry, but I don't know where I should stand. I am not used to speaking in front of people anymore outside of this box that's called Zoom. So with that in mind, I think I'll be doing a bit of walking around today in my talk from here to here, which is more interactive than just staring at the screen as normal. But yeah, it's wonderful to be here. Still can't believe that we made it here. And also with that in mind, I really appreciate all the fantastic science being done here, but also the hospitality, yeah, and the food.
And weather.
Weather is also fantastic. Yes, exactly. It's actually proper winter, so really appreciate that. So thanks, everybody. With that in mind, I'm going to talk about, yeah, I was told to talk a bit more broadly about programmed DNA elimination, but I guess the full title is a multidisciplinary approach towards understanding the evolutionary and developmental role of germline Soma genome differences. So it's a bit longer title, but I will come to that basically. But the idea is I will kind of keep this as broad as possible about this topic in general. What does it mean? And then, yeah, kind of hint at what we're doing in general in my group, but also why we're here and where we're going with this, hopefully. And I'm really looking forward to the discussion as well. So thanks so much, everybody, for being here. And I hope this pointer works. I guess what I'm going to talk about is not any kind of differences that we may have in our body in terms of our genomes. I guess all of us can relate to this. We have red blood cells that don't have a nucleus. So I guess technically that's also within individual genome difference, very predictable in a sense, but that's not what I'm going to talk about today. But with this in mind, I'm also going to just kind of show a couple things now to kind of just get us on the same page. What on earth is this, what I'm talking about? Why do we study this? I think you have all heard your fantastic colleagues talk about this before, but really for the bigger picture, what is that? And in order to know what something is, we should maybe first know what it's not. And I've been recently co-editing a special issue in Genome Biology Evolution where we looked at different types of within individual genome variation. So I'm putting this as an overview slide. I'm going to walk you through each and every of these mechanisms because then we get to programmed DNA elimination at the end of that. And with that in mind, first thing I would like to show you here is mechanism, I guess, that we can all relate to some kind of somatic variation. It can be any type of mutation that happens. It can also be somatic recombination, for example. So you have something that's, well, normal chromosomes in the germline, and then there's something different here in some of the cells or most of the cells. So I guess that's what we all expect. Maybe the most famous example that we can all relate to is the VDJ locus recombination. So that's somatic recombination in our own bodies. In this locus here, that is catalyzed by RAG1 and RAG2 genes and are basically, I guess, responsible for us being able to, well, fight this current pandemic with our vaccines as well from that perspective. So I think we can all relate to that. But there's a bunch more cases I would like to show you just from these different mechanisms, just for context. What is it not what I'm going to talk about today? Next thing is ploidy change. So within the same organism, we have different numbers of chromosomes, but these are just duplicated basically. So yeah, chromosome number increases in a predictable way. That also happens, by the way, in organisms that don't have a nucleus. So this is, I think, to me, the most fascinating example recently. There's some bacteria that actually have a lot of copies inside of this one cell. So it's a prokaryote. And then only the genomes at the end of the cell, the very tips of the cell, are those that give rise to the next generation. So technically spoken, you have actually germline and soma in one bacterial cell. It's quite a fascinating thing. And something to think about as well. What does it mean about, or definitions for what is a germline, what is a soma, what is conflict between different alleles in the same cell or in the same organism, basically, be it single cell or multi-cell, basically, from that perspective. Maybe another thing that's not often thought about in this context is just something outside of the nucleus. So this is basically any kind of heteroplasmy, different alleles or different variants that can have new plastids. In this case, mitochondria. So you can have maybe 2 different mitochondria from the germline. And then in different cells, you might have, well, 1 or 2 present or not present, basically. This is quite iconic example actually from these different clams. So in these clams, you can see that they have a unique way of inheriting mitochondria from both parents, and then they kind of compete in the same organism. And some tissues will have or will not have both copies or both variants basically existing there. Now we get a bit closer to the actual programmed DNA elimination, but still not. You might have heard of this. Uniparental genome elimination. So that's a situation where, for example, in hybridogens, so organisms kind of with 2 different sets of chromosomes from their parental species, they meet and, well, either during meiosis, but also sometimes during germline soma differentiation, some of the chromosomes or one set of chromosomes is lost from the parents, basically. And I think an interesting example here is from recent work on these carp gudgeons, this little fish here. They basically have these 2 sets of chromosomes, and then during germline soma definition, just one set of chromosomes is thrown out here. Just everything is put in the trash bin. And as a result, you have this germline soma difference as well. So also that, in a sense, is almost programmed DNA elimination. So I guess there was a long suspense of the stuff that's not germline soma differentiation in this regard in a programmed manner, because what I'm going to talk about from now on is only the actual programmed DNA elimination. So the first thing is really just fragments thereof. You could call it different ways. It's sometimes called chromatin diminution, sometimes called programmed genome rearrangement, for example. But I guess what they all have in common is that by definition, you start with a chromosome set like this, and then stuff is thrown out in red, and then you're left with a chromosome set that is just looking different in morphology and size. So something has changed in the chromosome morphology itself. I think quite famous examples are here these little ciliates, so single cell eukaryotes, then also some nematodes do that, and for example, also the lampreys. I will show some examples of details later. And just kind of to finish this overview to get us all on the same page, the second thing, and I think you've all heard a lot about that, are basically entire chromosomes, one or multiples that are eliminated during germline soma differentiation. So these are GRCs that we call them most often, at least in this audience here, I think. They're sometimes called also L chromosomes, limited chromosomes. But I guess, yeah, what they have in common is that they occur in very different looking animals. For example, these guys, different types of dipterans as well, so insects, but also these hagfishes here, which are somewhat distant relatives of lampreys as well. So I guess also that's another way of doing these things that we're doing. And with that, I hope it's okay if I move on to the next section of this talk, but just a reminder, have a look at this special issue, because especially the bacterial thing has been actually quite a surprise to my naive mind, because I don't think so much about non-eukaryotes very often, I have to admit. But yeah, let's now delve into more about what it actually means to do programmed DNA elimination. What are the different systems and what do we know about them at this point? Because I guess, long story short, that's not so much we actually know, except for a few systems where we have studied them in more detail. And then I mean, like the whole field, of course. This is not just us people here studying birds, but a bunch of other exciting systems as well. But really, getting to the point also, this is a nice overview, and to my knowledge, the only one where there's a phylogeny of all animals, at least, showing where these things exist. So this can be the stuff I just showed. Another thing that exists, for example, in mammals is occasionally the loss of a sex chromosome from the soma. But the point is, what all these systems have in common is that these differences are rather striking. So basically, there's a lot of DNA removed, eliminated, or entire chromosomes eliminated. So I would argue that what if this is only those systems where we can see them easily, so to say, and most of the other groups here in normal letters, they're just not checked for that, basically. So maybe with the iceberg metaphor, at this point, we just know the tiny tip of the iceberg in terms of what this mechanism does and where it exists. But with the tools that we're developing here, maybe we can also try to check that in other systems as well. I wouldn't say that as humans we have it, but who knows, right? Has anyone checked? I don't know. But yeah, so maybe with that, I should delve into the first system. I think that's almost, I think, arguably best studied in terms of programmed DNA elimination. It's these little guys called ciliates. So that's a single-celled eukaryote, which actually has two different nuclei. So one is a micronucleus, which is the germline, the bigger genome, the germline by definition. And the second one is the bigger nucleus, which has the smaller genome, and it's called the macronucleus because that's actually where all the kind of the working, the work of the cell is being done, the transcription, etc. So the idea is during the life cycle of this organism, the macronucleus can be replenished or replaced by a new micronucleus and differentiated again. So this is kind of just a way of going from generation through generation. And the interesting thing about this mechanism is that when you look at the germline of this organism, so the micronucleus, it is very, very messy. There's a lot of repetitive elements and fragments of different genes, different open reading frames in different orders, different scattered throughout, basically. And only through this process, transition from a micronucleus to a macronucleus, these things are actually being put together, really like a puzzle game. And then you have basically roughly one open reading frame per nanochromosome. So this is actually, in a sense, almost something like a splicing, but on a DNA level, kind of pre-transcriptionally, quite fascinating from that perspective. So yeah, just imagine 16,000 nanochromosomes. That's quite complex if we think about it this way. And the interesting thing about the mechanism is here that when we look at the genome in more detail, there is a certain way of predicting this. And as I said, this is programmed DNA elimination. So there have to be certain either genetic or epigenetic ways to redo that in the same manner each time we have a new generation. And the way this happens is that basically we have a domesticated transposase gene, or protein basically produced by this region here, so type of DNA transposon. And this is able to recognize and mobilize and chop out different parts, these intervening internal eliminated sequences, for example, and then re-scramble everything into these open reading frames here. So this is basically a cut and paste mechanisms where you just chop everything up into small pieces, put it back together, and then you have your nanochromosomes. And really, this is a very specific mechanism that is really derived of something that happens all the time in many different organisms. So DNA transposons are very common in different organisms, maybe not us humans, we don't have them anymore jumping right now as we speak, but a lot of other organisms do that. And the principle they use for that is exactly the same. So you have a transposase here, and it is recognizing specific interspersed or terminal inverted repeats, and then this way able to move these around, cut and paste. Same principle there. So the point of this slide would be that this is maybe a mechanism where these transposons and the host have been competing for quite some time, conflict, arms race, basically. And this might be a way for them to actually minimize this conflict, because you only tolerate the transposons in the tissue where it doesn't matter so much for you, because you don't do transcription from those regions. And then once you need to transcribe those things, you reshuffle, you get rid of the transposons, or the transposons get rid of themselves in a sense, and then you can solve some of these issues. So this is kind of the idea behind that. This might be kind of a conflict resolution of why this in ciliates might be existing here. It's quite a fascinating thing to think about. And yeah, might be looking a bit different in other systems. So I'm giving you a couple examples now because I think it's just beautiful to look at the diversity of these different organisms. Yeah, I think you all know this guy here, Ascarissuum. Maybe not so much beautiful organism, but the point is it's one of those very well studied for a long time. So its chromosome behavior through meiosis, mitosis, etc. Very well studied organism from that perspective. And it was noted quite some time ago already that the genomes look quite different between germline and Soma. And there's been, well, not so recent anymore work, but some of the pioneering work on animal programmed DNA elimination has been done in the system. And what people found when they assembled genomes is that you have some regions in your germline where you have DNA in blue that is shared between germline and Soma, and then red germline specific DNA. And then in the Soma, this DNA is not existing, but instead you have a telomere. So really it has been something kind of chopped into small pieces, and then something else has been added, telomeres, because that's the way to conclude the chromosome, of course. And there's sometimes a simple PCR from primers binding in these two regions here that tell you whether or not something is existing in this one tissue or not. So here we have the germline DNA, and this is clearly amplifying, but not in Soma, and vice versa here. So very simple way of also quickly verifying what regions are now adjacent to the telomere that were not before, because something has been chopped out in a sense. To my knowledge, I'm not sure what exactly is doing the chopping here, if that's also transposase mechanism or something else. I have to check more literature on that, but at least I'm not familiar with how this works exactly. But I think what is quite striking, because it's so well studied from a developmental perspective, this organism, here we know actually when the separation of germline and soma starts. So that actually begins in the earliest cleavage stages. So up here, we have only the germline. Here we have the soma, and each time you have an asymmetric cell fate. So you have one daughter cell that stays the germline, the other one becomes the soma by removing DNA. So irreversibly, you actually become soma, genetically spoken. And then afterwards, you have these different cell lineages emerging here. So that's quite an early distinction between germline and soma, simply through the presence or not presence of specific parts of your DNA. Quite a fascinating thing to keep in mind. And to my knowledge, it's not that well known for many other systems yet when exactly these things happen. So I think so it applies to the birds as well. We don't know for sure actually when that happens and what step is it actually? Is it the presence of DNA or the absence of DNA that does this kind of decision, of course? I need to mention this guy. It also looks a bit like a worm, but it's the deepest branch of the vertebrates. This is the lamprey. This is also has received quite some attention because it's the first system in vertebrates that has been studied this way. And here you can see one way of studying this. Basically, on this axis, you have basically the difference in coverage between two different samples. Blue being blood, so soma, and then red being sperm, so germline. So beware, this is flipped labels basically. And then you can see this is a normal distribution, and you have a local increase of coverage in these specific regions. So this means there are high copy regions on this DNA that's eliminated and single copy regions, and some of them might be not present at all in the remaining chromosomes. So this is, I think, quite a fascinating thing to keep in mind, because it actually applies to 20% of the genome that is eliminated this way, and quite a few hundreds of genes that are only present then in the germline and never present in the soma at all. So it's not just paralogs, but the entire genes that are eliminated this way. And I have to say, I got a bit surprised because the most recent work on this system by Jeremiah Smith and his group has actually shown that the whole story is even more complicated. So it's not just chromatin diminution, but also at least part of this difference is actually done through GRCs, so these germline restricted chromosomes. So it's a mix of both actually. So it's not always just one or the other. You can actually do both in some degree. This is, I think, quite beautiful work. This is some of the earliest cleavage stages here of the embryo. And you can see here in red, you have highly heterochromatic regions that are actually then the regions, the chromosomes, the entire GRCs that are being eliminated later on. So they're marked quite early on. And then when meiosis begins, you see they just stay here in the middle of the metaphase plate and then just get thrown out as these little micronuclei afterwards. And so that's kind of the resolution I think that we have now for lamprey, suggesting it's really a mix of these two mechanisms actually that's going on here, and really predictable in a sense in terms of the histone modifications, silencing histone modifications, and also DNA methylation markers. I think it's methylated cytosines basically. So these two things are in a sense also predictors for that. Also there, I think it's not exactly known what is removing the other regions in the genome, the ones that are not entire chromosomes. But it's fascinating to see also that the closer we look at these systems in the field, the more complicated the story gets, like with these lampreys as well. It's quite fascinating from that perspective. Yeah. So that's for lampreys. I hope it's okay if I jump to another system, which is actually now, again, outside of vertebrates, but insects basically, because I think those to some degree are even more crazy than the things I've shown you so far, at least when it comes to germline restricted chromosomes. One may argue if they may be even more crazy than the bird stuff. I will show you the bird stuff later in this talk, of course, but the idea there being, you have three potential independent emergences of these GRCs across different insects. So relatives of flies and mosquitoes, all the beautiful stuff that we like one way or another. So the first one are these Sciaridae, also known as fungus gnats, which in the germline have these two GRCs in orange here. Might look a bit more crazy. In this system, for example, here we have this is a Cecidomyidae, this is the gall gnats, which have, or gall midges, I think, which have a lot of GRCs, I think more than the rest of the number of chromosomes. It's quite massive what's being eliminated here. And the last system are the Chironomidae, or the non-biting midges, which have something intermediate in terms of its number of chromosomes here. So quite a fascinating system to begin with. And I think also there in that case, it's rather few species being studied in detail actually from that perspective, also genetically. So this is work that especially Laura Ross's group has been doing quite a bit over the last years. And I want to show you some points that I think we can keep in mind for that system, because it is a nice contrast to what I'm going to talk about in the rest of this talk in the birds. Excuse me. Yeah, I think it's easy on Zoom to not kind of have this problem with the microphone there. So the idea is with these dipteran GRCs is that as far as it's known so far is that they basically get eliminated from the Soma through just staying behind in the metaphase plate. So this looks very similar to what I showed for the lamprey as well. So I guess somehow they're able to be being differentiated or separated from the remaining chromosomes and then just eliminated this way. However, on the other hand side, when it comes to the germline behavior basically of these GRCs, and I'm just showing this for Skieridae, but it looks similar in the other systems as well overall, is that they kind of behave more or less normally in meiosis, except for the male side where one of the two GRCs is thrown out. And then both parents are giving basically one GRC each to the next generation. So as a result of this, you have kind of biparental inheritance of the GRC, unlike the system in the birds, which we'll talk about later. So with this in mind, I want to show you the one thing that's made the whole thing a bit more complicated. As I said before, whenever I think any of us look into more detail in one of these systems, it looks more complicated than we thought in the beginning. So I'm just going to leave you with this slide. I'm going to drink a glass of water before I keep on coughing into the microphone, because I think that's not so good for your ears. Sorry. So with that suspense, I think you all want to know what I'm going to tell about this specific plot now. The idea with this plot is to show you basically when Laura Ross's group was assembling now the GRCs from the specific system, what they saw is actually that instead of assuming from the previous slide that these two GRCs are the same, so more or less identical chromosomes, they actually are quite dissimilar from one another. So it turns out there are a bunch of GRC paralogs that are shared between these two GRCs, but the divergence between these is similar to the divergence that these GRCs have to the regular chromosomes, so the autosomes as well as the X chromosomes. So it doesn't really look like it's just a pair of chromosomes, but it's actually two separate ones, or that at least stopped recombining quite some time ago, because they're not identical from that perspective. Quite striking in this regard. And it gets even weirder when Laura's group was making a phylogeny of all these genes. In this case, they were not able to separate these two genes from one another, these two GRCs from one another, due to lack of information which gene comes from which specific GRC. But what they found is that basically when they made a phylogeny of these two these GRCs and the paralogs from the regular chromosomes, they saw that these don't belong together actually. So here we have the Skieridae, and here we have the Cecidomedia, and as you see that the GRC paralogs from the Skiera is actually down here. It's called the Rhodesia now, but basically the idea being is these are not closely related, and it's actually best explained by either ancient divergence of these two lineages and the GRCs thereof, but actually in terms of the phylogenies of these gene trees, it looks more likely to actually be a recent introgression of the GRC from one lineage to another. Not recent, but medium ancient actually from that perspective. So it is, I think, quite striking from that perspective, because that also these 2 points there were not really known in terms of what they mean. And I think I have to drink more water now. I'm sorry. So I guess the point being, we don't know for sure yet what exactly is the scenario here, because there's no genome for the other, the Cecidomeidae, but it's already looking way more complicated than was thought previously. And so it remains to be seen what is the exact history of these GRCs. Quite fascinating from that perspective. Also keeping in mind the Cecidomeidae have a couple dozens of GRCs, I think, so it's a bit more tricky to study those from that perspective. Okay. So with that, I think I'm finally going to move on to the birds. I think you've all been expecting that from that perspective. I'm going to talk about birds at some point, because that's also part of this collaboration of why I'm here. And as you can see here, this is a typical, I guess, snapshot of part of the chromosomes of birds, just the 8 biggest chromosome pairs. And then when you look at different tissues, you will find this extra chromosome. I'm pretty sure you've heard a lot about this from Anya Torgasheva's group over the last years, I think. And so this has been known for quite some time from zebra finch, but also from Bengali finch. I'm so sorry, I don't know what's happening to my voice right now. But that was it basically when we had started our project roughly 4.5, 5 years ago, basically. So the idea is for quite some time this has been known actually, but yeah, it was really unclear of how widespread this is and if it's just a kind of strange thing about zebrafinch and its relatives there. And I think you've all seen this very beautiful work from Anya's group showing that basically wherever you look, you'll find the GRC in songbirds. So it's most likely a very ancient chromosome, widespread in birds or songbirds at least, so at least 5,000 species of birds from that perspective. So I think that's quite striking in this regard. I think what they also found for the first time is that you can either have a large GRC, as I showed also just now for the zebrafinch, so very big ones, the biggest of the karyotype, as in the zebrafinch here. Or you can have a rather small one, a micro GRC. So there's quite some diversity in that regard as well, in addition to it being actually a very ancient feature of songbird genome evolution. So I think these two contrasting phenomena to keep in mind in this regard, of course. So I think that's actually, with regards to all the other systems I showed you, maybe the first system where we can have a rough idea of how widespread it is. So in this case, thousands of species. All the other things I showed you is, I think people looked at specific systems, but we don't even know for most of the species because they're actually not well understood from that perspective. So quite an interesting system. I think we can all agree on that. Maybe weird in different regards to the other systems that I showed before, but I think the remaining question, of course, that is this just a rare thing that is weird and doesn't mean anything, or is this widespread and means something? Well, it could also be widespread and mean nothing, of course, but the point being, either way, there's an interesting hypothesis put forward by Jeremiah Smith saying that, well, it looks like most of the deeper branches of vertebrates, including some of these cartilaginous fishes, basically, and then up here the songbirds, are having this phenomenon, different ways of it, so not necessarily the same way of doing it, but they have it. And for most of the other ones, we actually don't even know. So what if this is actually something that could be ancient, shared, or one way or another has existed for a long time? So it's hard to say, of course, if that's correct or not, but it just means we need to really understand more and more about these systems and dig more in detail in other systems. So maybe you'll find it in other systems when we start looking, basically. That would be the hope for that, basically. Okay. So without further ado, I think now stop the kind of extended points that I tried to make kind of for getting us all on the same page. What are these systems? What does it mean? What can we learn and what can we not learn at this point about them? And kind of give you some thoughts about where we are at with our kind of genomics bias perspective on GRCs from these songbirds. Because I think all the methods we establish now, we can hopefully also use for other systems and understanding them better in the future. And kind of really always seeing this from the multidisciplinary side. I can't emphasize this enough. We can't only do this on computers. We also need cytogenetics to really verify many of these points. This is very important to me to emphasize that it's not that genomics is now suddenly the solution to all of our problems. It actually brings way more problems than anything on its own, basically. So just for context here also, we spent a lot of head scratching and headaches over the last years trying to fix our genomics problems. But really, we're going to look at the system now in a bit more detail from a couple different angles. But I'm going to do this also, so I'm going to show them the methods and the results and a couple directions of speculations that you can take of what this might mean. Because at this point, I'm happy to say I have no idea what is going on with the system. I have a couple favorite hypotheses, but I think they're going to be very wrong. And so I'm just going to say, I don't decide now. I'm just going to show you what directions we can take these inferences, basically, because we'll see, right? In 5 years, I might look back and say, oh my God, what was I thinking? And I'll look at this on YouTube, of course, and see what I was thinking, basically. But the idea being, so we have the system, it's the massive chromosome, biggest chromosome of the karyotype, 160 megabases, so over 10% of the genome. So it's been studied rather well, even in the early stages before anyone was actually doing any sequencing. And this study here also was doing the first sequencing, 16 kilobases of the system as well. But the idea being, simply by it being such a large chromosome, you can actually look at it. And the idea is, or the default hypothesis is that, okay, you don't have it in the sperm. It's not inherited through the male line, only through the female line. Then you have a very big black box. As I mentioned before, we have no idea what's happening when. It's just looks like this when we look at the adults. So you have Soma never containing the GRC here and here. And then the germline looks basically either with one copy in males or two copies in females. And here's the first thing we can take away from that, depending on what is your favorite system or favorite background. You could say, well, this looks like a sex chromosome system. So what if this is an XXXO system? Because you have just one copy in males, two in females. So there's a lot of organisms out there that have XXXO chromosomes. Well, maybe not germline specific, but the idea being if this was a sex chromosome system, it would be a germline specific one on top of a ZW system, which is the opposite in the remaining body or in all of the cells, basically. Could this be anything? I have no idea, but I'm just putting this out here because this is something worth keeping in mind with many of the interpretations here, of course. Another thing I think that really changed the way over the last years was that instead of painful cloning, for example, or painful primer walking, PCR walking across the GRC step by step to get a few kilobases, we can actually get now at the genomics side of things. It doesn't make it easier per se, but we can get everything in our computer, and then we spend years just to figure out what is the GRC actually. But this is a project that we started basically almost 5 years ago now, and was started by Cormac, first master student in my group, and Paco, collaborator back then, and now postdoc in the lab, senior postdoc. And the idea is basically what we did was to sequence 2 genome assemblies from the same bird. So we take one Soma and one Germline, so liver and testes in this case, and then we do something that's kind of a way to get long haplotype information using linked read sequencing. So you make sure that basically more or less one DNA molecule meets one of these oil droplets, and at the end you get short reads, Illumina reads, that you can trace back to the haplotype of origin, basically. And this way, it allows you to separate haplotypes from different samples. Unfortunately, this technology is discontinued, so we're trying to kind of replace it with something else. But I think it's been quite promising and quite helpful at that point to really just first make sense of it. Can we actually find it when we sequence something? Because it is messy, as you will see over the next slides. The big principle behind that is that, well, for zebrafinch, we have a very nice genome assembly available through the field because people study vocal learning in the species. So we have here the A chromosome from the databases, and then we have our Soma linked reads. So we have 2 different haplotypes, and the same haplotypes we of course expect to find in the germline, right? Because this is the shared genomic components. This is just the chromosomes that are there everywhere. And then anything that's not found in one or the other is then called the GRC from that perspective. So these extra haplotypes we expect in the germline, if these have single nucleotide variants, SNPs, we can maybe also sometimes find them in coding sequence. So if that's the case, we can then say, this thing is actually a transcript from this specific gene, from this specific chromosome of origin. And in the same vein, if we can also do this on protein mass spectrometry data, so we could say, this specific peptide is coming from this specific chromosome of origin, the GRC. So keep this in mind when I show the results later. This is all something where we can separate the sequence of these different chromosomes, the different paralogs between the chromosomes. Otherwise, we can't say anything for sure, of course. So this is a key important thing to keep in mind here. And I think to summarize these points, kind of a lot of analysis taken together. The first thing I think that we notice is very much complementary to what Anya Torgasheva's group found, is that it is actually an old chromosome. So it was, I think, also for us very relieving to see that you found independently the same pattern, basically. So we did this only looking at one species, sequencing it, and then making a phylogeny of all the paralogs of all the genes that we can get from the GRC and the regular chromosomes. And when you make such a phylogeny, you can then say, okay, when these branch off quite far away from each other, this is like an old gene, a bit similar to what I showed with these fly, the Sciara GRC paralogs. So in our case, we have a couple genes that are at least common ancestor of all songbirds, true songbirds here, a couple intermediate genes, and a lot of very recent genes. So it's a rapid gene content evolution, but at the same time also really these old genes that pinpoint to the same ancient age of this chromosome as was found with the cytogenetic evidence as well. So I think that's quite a nice thing to keep in mind. And here we come back to the PET hypothesis. So if you're very interested in those kind of genes that are involved in cell differentiation and pluripotency, well, these 2 genes are involved in that. So would it mean that maybe the GRC has specific genes that, well, tell the cell to be a germline cell, or the absence of it means you're a Soma cell? Could be also a nice way to explain that, of course, but well, could also be wrong, of course, because we also need to look more if we're missing some genes at this point. It's just a subset of genes where we'll be able to do that. Another thing we can say is, well, the GRC is actually composed of genes from all over the place. So here's the thing that you can see here. It's just a circle spot with a placeholder for the GRC, and then each line connecting to where the chromosome of origin is for the paralog. So there's a lot of different chromosomes. Basically, most chromosomes, not all, but most chromosomes have kind of donated a gene, including a few genes from the Z chromosome. And if you think back now of the XXO system that I mentioned, for those genes that are sitting on the GRC, these would, in theory at least, be effectively balanced for its dosage now between males and females, because birds don't have a universal dosage compensation. So at least for those genes, that could be an explanation for why they're on the GRC. Could be complete random, of course, but it is, of course, with this in line with the sex chromosome hypothesis. That could be the way to explain this pattern. But really to keep in mind, depending on what genes you look at, it will look a bit different, or you will take different conclusions from it. Because another thing to keep in mind is that we have at this point around 115 high confidence genes, another 100 something that we are still verifying. So it's really just a subset. It could be completely wrong, the overall take home message from this, but with this subset, we can say the most strongly enriched terms are female gonad development and also some early embryogenesis. I think that fits very nicely with the idea if it's only female inherited, well, you can also evolve female beneficial function through a chromosome that's going only through the female line. That could be something to think about. Especially though, if we also think about the point that is expressed, but it's also expressed on the male side, as far as we can tell, both on the RNA level as well as the protein level. We have multiple genes expressed both in testes as well as ovary. And really importantly, it's the paralogs from the GRC, not the gene itself, but that specific paralog being expressed. So whatever this GRC is doing, it's doing something, both in testes as well as ovaries. And if gametogenesis is your pet hypothesis, of course, it could just mean that this chromosome is doing something in gametogenesis. Maybe it does. But really, as I mentioned before, depending on what genes we look at, we'll find something that will fit an interesting narrative, and that makes it so fascinating to study the system. Because another thing I want to show you is, well, what if this is just a B chromosome? I think, just a reminder, B chromosomes are these accessory chromosomes, sometimes called parasitic chromosomes, sometimes called supernumerary chromosomes. So no matter what they do, in the current moment, at least the emergence of it has something to do with meiotic drive through the asymmetry of female meiosis. And that kind of, I think, would be compatible with this inheritance mode here, right? If it's only coming through the female line, what if that's just kind of a remnant or still ongoing meiotic drive from this chromosome? That's why we see it. Again, I'm just going through the alternative hypothesis because it could also be completely wrong. So if you look at that from the previous work also with Paco involved, of course, when we look at the list of genes, we find there's one chromosomal, one paralog of a gene called SNPJ. That would be maybe quite striking in this regard. So because if you want to be a good GRC, maybe you have to make sure that you make it through meiosis. So you have to have meiosis associated genes. And SNPJ is also called CPAP. And that's actually involved in central assembly. Down here, you see those little links here, basically next to the microtubules here. So again, pick your favorite gene and your favorite hypothesis. You'll find something that would be interesting, because what if that actually plays a role, or at least it assists at least in making sure that the GRC is making it through female meiosis, because somehow it doesn't seem to make it through male meiosis, right? As I said before. Another thing I should mention, because I think for those of you who study B chromosomes, it might be just an interesting thing to think about with regards to very new data came out just a few weeks ago from this Mexican cave fish. This one has a very famous B chromosome, which seems to be now actually also somehow involved in sex determination. But this is, I think, to my knowledge, at least the first high quality assembly of a B chromosome in an animal. And here you have basically the chromosome model for this B chromosome. And each color is a different chromosome of origin. And here you have the corresponding color. So basically every single regular chromosome has donated regions to this B chromosome. And it's kind of the same pattern as I showed you before with the GRC in the songbirds. We don't know for sure how these things look like in their order, because we are working on a high quality assembly as we speak. But the idea is the principle is very similar as well, right? You have a lot of high copy genes, you have a lot of stuff from everywhere in the remaining genome. And you have a female bias inheritance from that perspective. So at least with these systems, I think it's something we can think about. Is that a possibility for why these exist? Is it the ultimate cause for it? Is it the proximate cause? Something to think about. I'll be curious to hear also your thoughts in our discussion later. Another thing to think about, because you have this differences in inheritance, could be, of course, sexual conflict, right? What if having a chromosome that only goes through the female line and not the male line helps you with resolving conflict? I mean, well, birds already have that because of the W chromosome, so maybe that's not so straightforward from this perspective. But again, just another thing I wanted to mention here in this context is we have a bunch of genes, and in this case, we have one gene under positive selection, a bunch of genes under purifying selection through selection scans that we did before. So again, the paralog on the GRC being under positive or purifying selection. And it turns out this one gene here, PUF60, is our only one that at this point with these analysis we can say is under long-term positive selection. I didn't think anything about this gene, but I'm putting it here because depending on what gene you want to find, you have these alarm bells that tell you something. Because this one, I had in mind the name of it because I didn't think anything about it until I saw this tweet by Judith Monk, who does fantastic work on guppies, where she said, well, look, the PUF60 gene, different alleles make you actually survive better as a male or a female. So this would be by definition a classic sexual conflict gene. So what is this doing on a GRC from that perspective, right? Again, pick your favorite hypothesis because you might find it from that perspective. So also there, this is something we need to think about more. Does this mean just there's maybe differences in expression or different alleles between GRC and the A chromosomes, and this way the GRC might actually be helping with that in the zebrafinches? Maybe. And the whole thing becomes a bit more complicated. As I said earlier on in this talk also, it seems like with every single system, once you start looking more deeply, it gets more complicated. I should mention here also, for example, Anja Torgerschewa's group and Yuba, you found, for example, variation also in the number of GRCs in the testes and ovaries, right? So it's not always just one or two, but there's a bit of flexibility in this regard as well. So it looks more complex the more species you look at and the more samples. And the same was also done now when Ivan, a collaborator in the Max Planck Institute from us, actually had a close look at the sperm ejaculates from zebrafinches, because we're looking very strangely when she was sequencing. She kept finding reads, so DNA reads that are looking very much like the GRC. So she went back and did a fish using a DPH6 probe, which is a highly repetitive region on the GRC. And lo and behold, every now and then you actually have a sperm nucleus where the GRC is actually inside of it. So we also verified this with confocal, different stacks there. And the idea is really every now and then, at least you will have a sperm that contains a GRC, contrary to the expectations previously. And interestingly enough, this number or the proportion of the GRCs that you find in the sperm seems to be something that's heritable, because we have some populations. So here in this case, one haplotype as a marker here, one population we have just one GRC inside of a nucleus every 100 sperm cells. And in another population, a different genetic background, we have on average 9% of sperm containing the GRC. Now that's something reproducible. Different males from the same family and also repeated sampling of ejaculates from the same males will always give you that result. It's roughly predictable in this regard. Very low efficiency, very high efficiency of the GRC to go through male meiosis for some reason. It is very strange, but also there, what does it mean for sexual conflict or resolution thereof? Because if now you have a GRC that's able to make it more commonly through male meiosis, does it just mean it can actually acquire male beneficial functions, or at least would be a nice vehicle for that? So also there, something to think about from that perspective. And then you may wonder, okay, well, if it's in the sperm, maybe it just doesn't matter because there's billions of sperm, so who cares? We also thought about that. So by chance, actually, was Paco looking at the data, and it turned out that also there we found some kind of indirect N-of-1 anecdotal evidence that I think suggests quite strongly that we are looking at something that is at least occasionally paternal inheritance. So this is a case in a Timor zebrafinch, which is one of the birds that Paco sequenced for the comparative genomics approach against different species that we're doing currently in the group. It turned out the first analysis showed us that, well, this is a Timor zebrafinch, but the mitochondria is exactly identical, 100% identical with an Australian zebrafinch. So it's a sister subspecies, 1 million, 1.5 million years away from each other. They should not be identical under no circumstances. And so when Yifan now went back to the regular chromosomes, the A chromosomes, she found that, well, most of the regions of the chromosomes are in red. So that's Timor variation, very clearly Timor. But there's some regions that are definitely Australian introgression. So it seems that this is more or less like a hybrid or a backcross of these two subspecies to each other. And when Yifan and Paco looked at the GRC regions, it looks like, well, we can't say for sure because we don't have a pure GRC from a Timor, but it looks most likely to be actually quite differentiated between these two samples. All the Australian ones have, for example, here, these 4 regions with no coverage increase, and then these regions are highly enriched in the Timor, or Timor quotation marks, hybrid basically. So what we think is going on here is that we have a disconnect between the mitochondrial DNA and the GRC, which is not compatible with maternal inheritance. It's not strict maternal inheritance. And really keeping in mind, this is a sample size of 1. So it remains to be seen, of course, if this happens very frequently, or if it's just, again, just a rather strange case and nobody cares about that. Who knows? But it was something to keep in mind as well when we think about what are the mechanisms at play here and what are the patterns that we see and how they can be explained, especially with regards to sexual conflict. And in this case, that would mean different GRCs are competing with each other in the same cell even. How does that work? How does that play out? So maybe one last point I want to make here is, well, I guess I can give you a spoiler that the GRC is very complex. So Paco and Simone, a new postdoc in the group, also are currently making a reference assembly with hi-fi reads, ultra-long reads, and omnic. And here's just one zoom into one part of the assembly. It turns out this chromosome has some regions. I mentioned this repetitive gene called DPH6. Some regions, in this case, 1.5 megabases aligned against itself. And you see, it's hard to see from the back of the room maybe, but it basically is this kind of little X-shaped and then tandems of X's basically. So it's a tandem of palindromes repeated over and over again, just one gene over and over again. And that very much reminds me, for example, of what is seen on the Y chromosomes of a bunch of different mammals, these ampliconic or palindromic genes as well. Doesn't mean that it has to be sex chromosome, but again, it's something really, we can see all the patterns that we see in many different other fields from that perspective. And again, it's really hard to say then what does the GRC do, or what is its purpose if it has one. So I think then I can conclude this part to kind of just say, well, the journey is basically going to understanding the why, how, and when. It's part of a generous funding that we just received from the European Research Council, but really also as part of this fantastic collaboration with Anja Torgasheva's group and Paul Mikhailovich. And so it's fantastic to be here to really add the cytogenetics perspective to this, because what we want to get at is really kind of these multiple disciplines to understand how the developmental aspect looks like, because without development, we won't be ever understanding how it's eliminated and maybe also why. You need to look at populations and how similar they are, what is the genetic variation there, and if there is deviation from maternal inheritance, as I mentioned, as we think might happen. And of course, seeing as if this is present in all songbirds, there's at least 50 million years old. So we should find some old genes, some younger genes, and see how this has evolved over time, because that again also might tell us what it might be doing, because the oldest genes might be more close to the original stuff that was ongoing 50 million years ago when this happened in the first place. So it's a fantastic team involved in that, and I'm just kind of putting these different names and photos closer to the direction. So we have Niki and Paco working more on development and species. We have Yifan working more on pedigrees and sperm evolution, and then Augustine and Simone more on populations and genetic variation. And I hope for this way we can really kind of understand these things in the next year. So I hope I can give you more updates next time we meet. So maybe to conclude then, what does it do? What does the GRC do? I think I gave you the answer that I have no answer for you. I just give you a lot of excuses for not giving you an answer. But what I can say for sure is that the GRC has a lot of high copy number developmental genes, and at least occasionally it will not be present, or it will be present in the sperm and then make it through the male line, occasionally, whatever that means. I think we can also say quite surely now it's present in all the songbirds. I'm expecting Luba and Anja to make a book about GRCs over the next years in terms of the morphology and all the different species, only 5,000 more to go. And also very rapidly evolving. So I think it's really beautiful to keep in mind what all of you will find when you started this more from a cytogenetics perspective. And then, as I said, you can pick your favorite hypothesis depending on what you want to see. And I think this is what's making it so difficult right now to decide on anything, because I might just tell you what's my favorite hypothesis, and I might be completely wrong for that reason, because you could find anything with regards to potential germline determination, antagonistic pleiotropy, B chromosome, meiotic drive, sex chromosomes, etc., from all the things I showed you so far. So very anecdotal evidence. And what I hope what we can do within the next few months is to kind of get to a complete gene content of the GRC for the zebrafinch at least, and then understand how old these genes are. And only then, when we know what's on it and what's not on it, can we then maybe say, okay, it's at least these 3-4 hypotheses, but not 10 different hypotheses, and then we can hopefully start testing those. That at least would be a very naive hope. And just kind of to put this in perspective for the bigger picture, when you maybe dream about GRCs tonight, I think we'll be dreaming about GRCs tonight. Why in the first place do we have to eliminate DNA? Assuming that we humans don't do it, why are we okay without that? So is this just something that helps with removal of repeats, minimization of conflict between those elements? As I said, for ciliates, that's something assumed. Is it removal of germline expressed genes? Because then you don't have to regulate them if they're not there in the soma. They will never get misexpressed, so you can't get cancer from that, etc. This is something that's been implied from some of the genes found in the nematodes, for example, in lampreys, and it's also tempting to assume the same for the GRCs in songbirds. And really this idea, back to this point of the iceberg, how much are we just missing, and how much of this is just being odd versus the rule across the tree of life? I think we're just starting to anticipate how truly variable genomes are. I think many things were known for decades on a cytogenetic perspective, and then genomicists came and said, nah, we don't find that because, well, we can't study it. And now we can start studying these things, basically. And really, as a last point there, what does it mean for cell fate? So if you don't do rearrangement or removal of your DNA, so you go from here, zygote, and then your chromosomes go on here, and everything looks the same in somatic tissues and germline, you have to do everything through gene regulation, right? So this is maybe the default that we assume for ourselves as well, and cell fate has to go to something, maybe sometimes with germplasm, etc. But what if that's maybe a shortcut to do if you have rearranging species, because they're can just say as soon as you eliminate this DNA, you are by definition the germline, and then there you already have kind of this dichotomy on cell fates. Might be an easier way. This is something that also Jeremiah Smith has been proposing as a way to explain why this might actually just simplify this whole situation here. And I find that very tempting, but again, we need to test it, of course, if it applies to all the systems here. I think this is mainly based on lampreys. With that, I want to thank a lot of people. I put the key people on the slides, of course, but everybody's here. I think you notice also this logo, people here. Fantastic also to have this group here of people, and funding, of course, and consortia for providing somatic genomes. And yeah, very happy to take questions. Thank you so much.
Okay. Questions?
So I have two questions, and the first one is, even in human, I think some time ago, it was a work when they said that if we consider a heterochromatic region, then we can find several lines. And these lines, they appear very, very early in embryonic development. So it happens something to heterochromatin. So some parts are getting removed, if it's something similar maybe going on there. Yes, yeah, they found it in human because human is so well studied back, I think more than 10 years ago. But I don't know if they validated this data somehow. Really
Not to my knowledge, no.
Okay, and the second question is about the germline cells. Do you think if somebody has compared extra chromosomal DNA in germline cells and in somatic cells, and maybe there is something we can see the chromosome, something what we can see in microscope, but maybe something really small which we cannot estimate somehow, and when we do genome sequencing, we always put it into the rubbish bin and never check it again. And maybe something like pre-mutation for the appearance of germline restricted chromosomes or big chromosomes on that level.
Thank you so much for these two questions. I should maybe add one more thing for the first question. I think, to my knowledge, it's not been checked. I mean, I'm not too familiar with the human genomics literature, but I think the point being, as you said, with the rubbish bin, many of these regions, if they're so repetitive, it is often impossible to say, well, is it now 5,000 copies or 4,000 copies? Also that is, always within kind of coverage, noise, etc. from that perspective. So I think, yeah, it could be very well possible that in many, if not most organisms, there is a lot of variation, at least in those high copy number regions, and we just don't have the tools to reliably actually predict how different they are between tissues or even within tissues also from that perspective. And for the second question, that's a very good point. I mean, the extra chromosomal circular DNA, this has been one of those mechanisms that we've been discussing in our group, and Simone, one of our postdocs in the group, she's actually planning right now experiments to do just that, because we don't know for sure how this would work otherwise. We looked into different mechanisms of how genes or regions from other chromosomes can be copied over to the GRC, and we can exclude at this point, for example, transposable elements. It's at least not the most likely playing a role here. So I think it's really a mix of understanding this from data types, like, as you said, these extra chromosomal circular DNA, plus also the population perspective here. What I hope we can check for is whether or not there are genes that are present only in some individuals and not in others, because they might have been added yesterday, so to say, just came in. And then when we look at the sequence characteristics, there are the breakpoints, we can kind of rule out what it's not, at least what are the mechanisms playing a role here. But I think, long story short, it still doesn't solve, of course, the issue of how it emerged in the first place, right? I think maybe Paco can give a better answer for that, but my naive assumption would be I find it most plausible to just start with the same mechanism as for a B chromosome, just something like an aneuploidy or some well, extra inheritance of, you suddenly have a meiotic driving element one way or another, and then once that's stabilized, you can just add more stuff afterwards through the mechanisms that we just discussed. So, but also there, it's always hard to say. It's so many steps involved that it doesn't. None of these seem to be particularly parsimonious. I don't know if that answers your question.
What about traffic between A and GRC? Do you think it's unilateral or bilateral? Any idea about it? Can we check it? Can we distinguish these two ways?
Yes, I think we can. So I mean, not yet, but we're working on that. I think the idea is once we have in-depth assemblies, so the thing is really being able to separate all of the regions on the GRC
Yes.
from the A chromosomes is right now the limitation. There's some regions that we can separate very clearly and others not so much. Once we have that, we can make phylogenies across populations as well as species to see, okay, well, if it sits at that part or that part of the tree, you can say if it was that direction or that direction. I guess by default, the null hypothesis would be it goes from outside to the GRC because at least so far as is known, I think there's no other birds outside of songbirds have the GRC.
Yeah.
So that means there would be a new trait of the songbirds and everything would have come there, but I would not see why it can't go back as well. That would be plausible, of course.
Alexander, thank you for the exciting talk. Coming back to the first part, I was interested in this phenomenon where you have a genomic splicing in this unicellular organism. So there are examples of some highly evolutionary, highly developed species, like some fish, Medaka fish. I think it has also like the genome which doesn't have lots of introns, like mostly exons in there. What are the speculations, like current speculations? What is the function of such like intronic elimination during evolution or whatever? What does it bring to the species?
Oh, wait.
Yeah.
Okay.
I think it's a very good question. I guess I will start with a blunt answer. I don't know for sure, but maybe to differentiate the point here. So as far as I'm aware of from Medaka, I'm not 100% sure, but I think what you're relating to is that they have a rather small genome, so they have had a lot of deletions, and that's why introns look rather small and also intergenic.
Not during development and just during
exactly.
the evolution.
Exactly.
As an evolution of most birds. They lost a lot of, okay, sorry.
Exactly, that's what I wanted to get at, because I think we have to always differentiate the mutations that happen within the organism, and then anything that happens from Germline to Soma, of course, will be lost as soon as the organism dies. But there's a lot of dynamics also happening from one generation to another. So either through meiosis as well as early development, that's many of these transposable elements, for example, are jumping and or you have, for example, non-allelic homologous recombination. So you have regions that are identical. Hope I'm not going to drop my microphone. You have identical sequences in different spots, and during meiosis they don't recombine with the other chromosome but with each other because they're identical. And this way, one way to resolve that could be, for example, to make a deletion and then this way shrinking. So we've done actually in my group we've done some work on that in the past, and what we do see is a correlation when you have more transposons jumping. A default, at least in birds and mammals, not too much in crazy genomes like salamanders, but in those organisms here, birds and mammals, if you have more transposons jumping, they can form these loops and then delete a lot. And so as a result, you have also more deletions and you actually shrink faster. So this, I think the second aspect here is that whenever you look at different organisms, the genome size and the genome organization you see is kind of a result of this interplay between DNA gained over time, for example, through transposons, and DNA lost over time through weird resolutions of these homologous sequences. And but it kind of relates back to the same point as you made them within the same organism. I think in both cases, it might sometimes be actually beneficial, at least not so detrimental, to have smaller introns, for example. So then it's hard to say then if there's strictly spoken, strict selection on it, but in a sense, if one way or another you can minimize this conflict between the transposable elements and their hosts, for example, by splicing with ciliates, as I said, or in this way also by resolving recombination events through deletions, you actually remove also the transposons in there, right? So this is another way for dealing with that, and so the transposons will be only left behind in very few regions of the genome, as we actually see in the birds also. They're sitting mostly close to centromeres.
They lost a lot of introns.
Exactly.
They lost even parallels.
Exactly. Yeah. So
they have twice as smaller genomes than, for example, mammals.
Yeah, but close species. Very close species.
Sorry?
Very close evolutionary species could have like one could have longer introns.
exactly.
More questions? Yeah, now, first, we are talking about that's a very fundamental question. You are talking about germline and Soma. How and when they divide.
I mean, I think I would naively speak about
saying birds. Well, let's restrict that to birds.
To my knowledge, it's not really known when exactly it happens. It's just more at certain point. It's easy, not easy, but it is possible to differentiate the primordial germ cells, and that's a rather late stage already. But whether that means that the germline soma differentiation happens, I think that's shortly after gastrulation, for example, or if it happens quite early on, maybe even as early as I showed for the nematodes, the very earliest cleavage stages. I think that's, to my knowledge, at least not known in birds, for example.
Do we know the mechanism how it happened? In any species, or it's just my ignorance?
I have to admit absolute ignorance for that. I don't know. I have to admit I don't know. Yeah, exactly. I mean, I have to admit also with that said, I would hope so if the GRC, maybe now is my wishful thinking speaking. So again, this might be just me being completely biased, but if the GRC has anything to do with germline soma determination, we can use the GRC to say when exactly is that happening. But if it has nothing to do with it, of course, we will never find out from that perspective, I guess.
So the GRC is the tool to solve this, problem. Yeah, look at that.
Maybe.
Zach, I don't like the picture of Smith you showed because it's it's extremely misleading.
Which one?
The one with that one. I don't believe that all lampreys are have this programmed elimination, or all hagfishes. They studied, but they studied about two or three of them. Is it?
I think I'm not sure for lampreys, but I think for hagfish the last read was it's around a dozen species of hagfishes, and each of them has GRCs, and it's also each of them has a different number of GRCs. For the hagfishes at least. For lampreys, I don't know actually how many have been studied there. Yeah.
And all about this, what's that?
Exactly.
This mosquitoes.
No,
they also it's very, very small branch. It's not like 50 million, 500 million years ago. I think anyway.
If I can answer on the mosquitoes or in general the dipterans, I mean, I think it looks. It's at least it looks like it that there are a lot of species where that don't seem to be GRCs. Can be loss or independent gain in these three other lineages, that's for sure, or two tiny ones. Exactly. That's very possible.
Okay.
Again, thanks for the talk. I was kind of wondering, you mentioned that in birds and at least in one species of fish, there are genes from different chromosomes in GRCs. And I was wondering, are there any GRC-specific genes in GRCs? And if so, are there what percentage of them?
Yeah, so I think I didn't mention that explicitly when I went to this one slide here. Gonna just a moment back. I have the gene ontology enrichment. One more. This one. So all these genes, 115 high confidence genes, are all with a paralog on the regular chromosomes. So for those, I guess there are different degrees of being differentiated or diverged, but they're all rather similar in sequence identity. And then there's another 130 genes, I think, that are also all having one paralog on the regular chromosomes. We have one candidate now, but I wouldn't bet on it too much because so there is one candidate where we might have no paralog on the regular chromosomes. However, it is one of those genes that belongs to this category of hidden genes. I don't know if you're familiar with this discussion in bird genomes. So even some of the regular chromosomes are very difficult to assemble, the smallest chromosomes, the microchromosomes. And the smaller they are, the more the GC content, the higher the GC content, and they often don't show up at all in genome sequencing. And so I would want to wait that we can be absolutely sure that it is not just hidden on those microchromosomes, but we can differentiate whether or not is truly only present on the GRC or not. So I would just kind of carefully wave my hands on and say there might be one, but we have to check this further. But that would be basically one out of over 250 or so. And so everything else seems to have a paralog, and that is rather strange, in my opinion. Yeah.
Thank you for a very interesting talk. I have a question. If we talk about programmed DNA elimination, can we estimate the degree of regulation of this DNA elimination? Is it hard or soft? Is there a room for variation between tissues, for example, or anything else? How is it regulated? I wonder.
Thank you so much for this beautiful question. Yes, I guess at least with the GRCs, it seems to be a bit more discrete, right? You have a GRC, you don't have a GRC. At least that's what we can say so far. On the other hand side, I guess we would have to also double check if there's fragments of GRCs floating around. We don't know for sure, I guess, if I haven't checked for that. But it gets a bit more fuzzy or a bit more, I guess, dynamic with regards to the less restricted to one chromosome situations. Like with the ciliates, I think there is a bit degree of variation that can happen. I'm saying this as somebody who doesn't study the system, but I think there is a bit of plasticity in this regard. And I remember also some earlier work from Jeremiah Smith on the lampreys, where I think they measured the genome size of different tissues of lampreys, and also they varied quite a bit as well. So it could at least reflect some degree of differentiation, that it's not a one like snapshot that it changes to one another, but it maybe happens across multiple stages even, and as a result it might have between tissue variation as well, or maybe even within tissue variation, which I hope, I guess, in the next years when we get better single cell sequencing technologies, we can actually get at that, to know how different they are.
Yeah, in fact, we really don't know about Soma. We know it's not in bone marrow, we know it, yeah, but we didn't really examine it, all tissues to exclude.
Exactly. So I have a follow-up about like tissue specific effects. Maybe in between, like it's even cell type specific could be something like this. If you have a dosage effect, for example, you have one cell type which does have the chromosome, another doesn't have, and then you have a competition, and one gene, like one cell has like double dose of. Do you know something like? Some speculations about. I know that there are some epigenetics and like extracellular signaling mechanisms to do this, but maybe with genome.
To be absolutely honest, I think we need to wait for some kind of close to single cell resolution with that. So what we're trying to do now, we actually have Nikki, one of the students, is currently, as we speak, basically sampling embryos, different stages, and also gonads, different stages to get at that. We want to try this technology called spatial transcriptomics to really see if this way we can get kind of both histologically as well as RNA sequencing resolution of how different these are. Because if we also keep other slices, we can then check, for example, for the presence of the whole GRC or fractions thereof in different tissues that we don't currently at least assume to be exhibiting GRC. So I kind of hope this way we can actually check this point, that maybe it's not clear cut, but every now and then you have fractions thereof as well.
Yeah, but well, look, we have very indirect and very preliminary data, but Luba demonstrated that 20 hours after conception, the embryo contains about 5 to 20% of cells contained GFC. All the rest are left are without GFC. So that's likely it's more as Sasha formulated, it had control. Yes or no? Yeah, well, but we have to go more in depth. Yeah, more questions?
Can I add one thing to the hardness of this? Huh? I think the hardness of this distinction depends also a bit on if this is a single elimination event, just one or multiple times, right?
In one progenitor cell, exactly. Or in just exactly. No, I don't think possible. No. There is no one cell which nobody knows.
I mean, if you think back of the nematode example I showed, that's multiple bifurcations where just one of the daughter cells is Soma, and then independently you have the removal there. So I guess there could be quite some variation between those as well in terms of the elimination, I guess.
Yeah, what now? What's the sense to have two GFC in females and one in males in before meiosis? And what's wrong with GFC in sperm? Why they took all these troubles to get rid of it? Why it's tolerated in eggs but not in sperms? What's the point? And okay, yeah. And at what time these sex differences are established? What time of development?
All these steps, I think, are part of the black box that I showed in this diagram from the E2 adult paper.
Okay.
This is exactly the thing. I think it's really just we only know the two states, zygote or before zygote, and then the adults. And as a result, I think it's really unclear. Does it just mean there's some kind of endoduplication, for example, a non-disjunction that leads to 2 copies versus 1? Is it something else ongoing? I mean, if there's, for example, room for paternal inheritance every now and then, could that help to have 2 copies? Well, if that was true, that would have to have 50-50 of the times, right?
Yeah,
and that makes no sense. But you know what I mean? Like it's
male and female have to be the same.
exactly.
Well, the tragedy of bird meiosis is meiosis and early development, that everything happened in the egg, and the egg inside the duck. And what else? The duck inside something else. Yeah. So it's all right with ducks. They can kill it and eat, but you cannot kill all these poor zebrafish. Hundreds of pregnant zebrafish you cannot kill. Just to examine, different stages, the most interesting stages of chromosome segregation. That's the tragedy. Yeah. Okay. Any more questions? Okay, thank you very much, Alex. That was really a great talk.
Thank you so much. Спасибо.
So, next Wednesday, Francisco will tell us about satellite DNA as a tool of cytogenomics, and on Monday, in interlaboratory seminar of в общем, в пятницу, в понедельник на межлабораторном семинаре в 3 часа Алекс будет рассказывать про что? What are you going to speak about on Monday?
Microphone, yeah. I'll talk about the W chromosome of birds, so sex chromosome evolution and retroviruses that are sitting there and what it means for physiology and mutations of birds.
And Francisco will talk about how to determine what is it in Jersey. Yeah. So see you there and here again. So goodbye. Thank you.